The result of all these studies, right from 2017, we published in 2022 an article in Theoretical and Applied Genetics on host plant resistance for fall armyworm management in Asia. What's the relevant status and prospects for both Africa and Asia? One important thing that we need to do whenever you do germplasm screening for host plant resistance is not only take into consideration the foliar ratings. That means right from score 1, which is unaffected completely, no even pinholes on that, all the way up to score 9, which means it is completely damaged at the foliar level by the insect, severely damaged. Not only foliar responses, but also the ear responses need to be recorded under artificial infestation. Right from score 1, that means absolutely no effect, to score 9, where all the kernels were eaten away by the fall armyworm, because the insect can bore through the silk as well as through the ear husk and then cause extensive damage. This data recording is very critical. Don't look at only foliar data, but also the ear data. If you look at the foliar data, you can see CML 71, CML 338, CML 444. CML means CIMMYT maize line. CML 574, CML 338, CML 71 offer excellent even foliar responses under artificial infestation compared to, you can see here, a drought tolerant inbred line, which is highly vulnerable to fall armyworm, or a CML 395, which is an elite line. So using these robust protocols, we identified a set of inbred lines which are excellent against fall armyworm and distributed globally: Africa, Asia, Latin America, North America, even Europe, all the way up to Australia. These are the number of institutions, and these are the number of countries. So you can see 92 institutions from 34 different countries have benefited out of this germplasm. The second most important thing to do is, once you have a highly promising hybrid that is identified through choice trials, where hundreds of entries are grown within the screen house, you have to also set up a no choice trial. No choice trial means one big screen house divided into 2 or 3 different partition, and each of this chamber will be growing only one entry, and each and every plant in that chamber, that means even 30, 40 rows of a hybrid, are infested at the foliar and reproductive stage, and then you record the response. That means there is no confounding effect of any other germplasm, any other entry. And under such conditions, how does your entries respond? Either the insect has to survive and eat, or it has to perish. So under such conditions, you can see here the fall armyworm tolerant CIMMYT hybrids absolutely clean in such no choice trials, compared to susceptible commercial checks which are severely defoliated. You will hardly get even less than 1 ton per hectare. And even if you get some grain, it will be highly contaminated with insect eating. On the other hand, you see here how the fall armyworm hybrids are performing. So, and then when you look at the yield data from those no choice trials, this is how clean are the ears compared to the insect eaten ears, if at all they are there. First of all, the yield is severely penalized. Second is, even if the yield is obtained, the grains are very much prone to susceptibility to various other fungal infections. So through this strategy, in December 2021, we released the first set of fall armyworm tolerant white maize hybrids for Africa. And now Kenya, Ethiopia, South Sudan, Zambia, Malawi, Ghana, and Zimbabwe have not only already released these hybrids, but they are now commercializing through seed companies. Similar national performance trials are ongoing in several countries in Asia. My request to the Philippines colleagues is to get this germplasm, evaluate it under local conditions, and start releasing fall armyworm tolerant native genetic resistant materials. So we have now, as I told, intensive screening being undertaken at CIMMYT Hyderabad under fall armyworm artificial infestations. Already a total of over 138 and inbred lines identified to be tolerant after rounds of screening. Over 30 breeding populations with Falamiwarm resistant donors have been generated and used in our breeding programs. You can see here again our resistant hybrids from Cimitaceae pipeline compared to the susceptible commercial checks in some of our Asian countries. So it is really quite strong, almost 5 to 6 tons per hectare difference between a Falamiwarm resistant hybrid versus a susceptible hybrid. The question that we need to ask next is what causes this native genetic resistance? A range of characters, right from morphological traits, physiological traits, biochemical traits, and even molecular basis of native genetic resistance is increasingly being understood, including proteins related to, for example, cysteine protease, or beta caryophyllin. It's a terpenoid molecule or a secondary metabolite. So a number of genes, including those in terpenoid biosynthesis, flavonoid biosynthesis, have been identified. So it's not only non-preference, but also antibiosis that you see. Non-preference is because of leaf thickness, high trichome density, tight husk cover. Those are typically contributing to non-preference of those hybrids. And these secondary metabolites, including phenylpropanoids, caryophyllins, etc., they cause antibiosis. So we are now increasingly understanding the basis for native genetic resistance to Falamiwarm and what inbred lines offer what sort of mechanism. For example, CML71 not only shows good antibiosis, but also good non-preference for feeding. Whereas CKSBL, that is a Simit Kenya stem borer resistant line, showed excellent antibiosis on leaves, besides non-preference for feeding on leaves, silks, and ears. So several national partners have started using these materials in breeding for native genetic resistance to Falamiwarm, including colleagues in Ghana, including colleagues in India, Zimbabwe, and the list is several countries. So I'm just showing you glimpses of this. Coming to the BT maize, there are, of course, BT maize projects which are implemented in Africa in collaboration with Bayer, with African Agricultural Technology Foundation in 5 different countries in Africa. BT maize has been already released in not only in South Africa, but also in Nigeria, and recent approvals are also coming up in Ethiopia. So this is what we call as the Taylor maize. It's a Monate 9034 BT maize, which can offer resistance to Falamiwarm as well as stem borer. In Asian context, BT maize has been in cultivation in Philippines since 2003, in Vietnam since 2015, in China it has been recently released, in Pakistan they are being tested. So there are a number of countries in Asia too which are beginning to grow or give approvals for BT maize hybrids. But one thing one has to be very careful about when we are talking about BT maize is insect resistance management. Already we are seeing in China and in many other countries insecticide resistance characteristics in several of the Falamiwarm populations that are invading this country. So robust insect resistance management has to be a key component of an integrated pest management strategy, especially if you are relying on BT maize. You have to prevent or mitigate the onset of resistance. You have to better understand the population dynamics of Falamiwarm and resistance profiles across the region to guide our response strategy. But if I have to manage Falamiwarm, whether it is in Africa and Asia, in a much better way, I won't rely only upon BT maize. I will rely on native genetic resistance too, because native genetic resistance is a polygenic trait compared to a BT maize, which is typically monogenic or oligogenic nature. So together with Bruce Tabashnik, the guru of insect resistance management, we came up with an article on how can we sustainably improve control of Falamiwarm. So where we recommended clearly use BT maize with other pest management methods, including native genetic resistance as a part of your IPM plan. Avoid single toxin BT crops. go for BT maize pyramids and things like this. So I would certainly prefer a BT gene to be incorporated within a native genetic resistant background rather than a susceptible background. So the resistance allele frequency of fall armyworm are changing for CRY1, CRY2, VIP3A. There are patterns of cross resistance. There are fitness costs associated with resistance. So that's the reason why new BT toxins are also being probed by several institutions within Asia. One more aspect is genome editing for fall armyworm management. The fall armyworm Spodoptera frugiperda has been sequenced. The genome has been sequenced. Genes responsible for various critical reproductive functions have been now identified in fall armyworm. So this would provide an opportunity to create fall armyworm populations it's like for providing mating disruption and release that in environments. That could be one potential strategy. A number of such functional genes have been characterized in Spodoptera frugiperda in recent years. Look at this paper in 2024. An array of genes have been identified affecting different aspects: chemosensory regulation, recognition, biosynthesis of female sex hormones, pheromones, sex determination and reproduction, growth and development, spermatogenesis. There are a number of gene functions that have been identified. So in summary, friends, both conventionally derived and BT-based resistant maize varieties have their own importance in fall armyworm management. We do need to intensify breeding activities in South and Southeast Asia for developing elite maize germplasm with fall armyworm resistance and fast track the deployment of fall armyworm tolerant elite maize hybrids. In countries like Philippines, where BT maize deployment is happening, one must give due attention to insect resistance management. And it is certainly possible to achieve synergies between host plant resistance and other IPM approaches for eco-friendly management of fall armyworm. Thank you so much.
Thank you very much for the presentation, Dr. Prasanna. Should we head to Dr. Christian?
Sure.
Or okay, so Dr. Samira will be the last presenter. So let's proceed to our fourth speaker. We have with us Dr. Christian Thierfelder. He is a principal cropping systems agronomist at CIMMYT, specializing in conservation agriculture or CA systems research across Southern Africa. He led CA research and implementation projects in Malawi, Mozambique, Zambia, Zimbabwe, and Namibia since 2004. His work focuses on adapting CA systems, cultural practices, and appropriate scale mechanization to the conditions of smallholder farmers. Ladies and gentlemen, let's welcome Dr. Christian Thierfelder.
Hello, everybody. Thank you so much. Can you hear me properly?
Yes, sir, loud and clear.
Wonderful. Then let me continue with my presentation. I'm not a fall armyworm specialist, I must say, but I specialize in cropping systems research. And that has a good reason in this presentation. So when we observed in 2016-17 the emergence of cropping issues with fall armyworm, as you can see from this map, we were puzzled by differences in cropping systems, how they responded to fall armyworm. And that was an interesting aspect that we wanted to explore further. Because, you know, for most smallholder farmers that I'm dealing with, one of those very highly sophisticated control options, BT, are probably out of reach. But cropping systems, if combined with, for example, resistant or tolerant maize varieties, could be a combined control strategy. So when the pest arrived in this area, the immediate reaction by governments was chemical control. They panicked. They all bought pesticides. often insecticides that did not even work for fall armyworm and were not actually released for that purpose. So some of the responses were not very encouraging. The damage was horrible in some areas, and some of the yield penalties that were portrayed by several researchers were enormous. Yield losses between 20 and almost 70% were postulated, but the reality is actually slightly different. Control methods based on agronomic strategies are probably more affordable for resource-constrained smallholders. They represent low risk for health and the environment and can be easily adopted. So our objectives in this work was, first of all, to estimate fall armyworm damage in smallholder maize fields outside research stations with real smallholder farmers in the field, following these rigorous scouting protocols. Then understand factors influencing the fall armyworm damage, identify natural enemies, and also identify different cropping systems that could help controlling fall armyworm. And we're building on the knowledge from organizations like ICIBE, for example, that helped us really design climate-adapted systems for our situation. And that was mostly Southern Africa. So what we did in a first step, we did some field assessment in rural districts of Zimbabwe in 2 cropping seasons to really understand the damage, severity, etc., of fall armyworm in smallholder farmers' fields. We use those zigzag protocols that have been shown before, looked at number of plants with leaf damage, number of plants with frass in the world, damage scores that have been also looked at, and also scores on the crop itself. We then used a technique that CIMMYT developed over the years for a rapid yield assessment, looking at crops that we harvested from specific areas that we photographed, used a computer to evaluate how much yield we would get from those using specific techniques developed by my colleagues from the maize program, and then assessed what would be the yield of those systems. So what we found out in those 2 years where we did this research in 2 different districts, Chipinge and Makoni, that we had leaf damage in the range of 42 to 55% in the first year, and then 75 to 49% in the second year, and damage scores around 3.7, 3.8, sometimes a bit more, 5.3 in the second year. And then what was very interesting, we then looked at factors influencing fall armyworm damage through a very thorough survey that we did with the farmers, asking them about their cultural practices, what they do in their fields. We found, interestingly, very little effect of those pesticides that were applied at that time. Now we have better pesticides that can control, but at that time, no effect. Even the presence of hedgerows or pumpkin intercropping did not lead to any positive effect on fall armyworm. Instead, for example, pumpkin leaves could be a hiding place for the moss or hedgerows from where they migrate into the maize stands. And some maize varieties showed a little bit of control, some absolutely not. And that's actually when we've become very, very grateful for the work that Prasanna and his team did. Then rotations with legumes or fallows were very beneficial, especially with legumes. Conservation agriculture, a cropping system based on minimum soil disturbance, crop residue retention, and crop diversification was very effective. A balanced fertilization was good. Application of composts, of frequent weeding, or early planting were medium to well in controlling fall armyworm damage. We also tried to estimate the actual yield loss that we would get with different infestations. And what we found in those 2 years, only some 12% to 22% yield loss from the actual work. And when we just looked at the crops themselves, it was slightly lower. So those huge numbers that were initially put forward did not apply for our situation. That could be several reasons, but this was not as dramatic as everybody thought it would be. We then followed on to establish on-farm trials, replicated on-farm trials with different, what we call climate-adapted push-pull systems. Push-pull is a system that was developed by ICP. I'll talk about that in a minute. So we used crops that are common in the farming systems of Southern Africa and intercropped them with maize, with brachiaria hedgerows and without brachiaria hedgerows. And then we looked at what would be the effect of this. The push-pull system is a system basically developing two control mechanisms. One is this push that comes from a legume called Desmodium uncincatum, and the pull from a grass that attracts the fall armyworm out of the maize stand and helps with this double action of push and pull to control the pest. We interestingly found several legumes that reduced the damage. For example, cowpeas as an intercrop, or lablab or velvet bean, even groundnuts reduced the damage scores in the field. And that's actually what we wanted. We wanted crops that are available and are used by farmers in the fields to help us control fall armyworm. We also had traps in the field to really find the enemies or the positive insects that help us control fall armyworm. And it was interesting. Spiders, parasitoids, ants were measured in those two districts, and there were differences as well. In some districts, for example, ants were much more pronounced than in other districts. And some of the cropping systems also responded better to those, and we found more of them. So that made us think, what is actually helping us in controlling fall armyworm and what is actually not helping us. So ants were identified as one agent or one insect that really helped us in reducing the damage. The more ants we had, the better it was for the maize. Also parasitoids, although we did not establish such a clear relationship with those. It's a complex issue. What you need is animals, insects that attract, for example, the natural enemies. Here we have the alternative prey. These are insects that are good for attracting ants. And then we have the enemies. In this case, Formicida is ants. The more ants you have, the more we can actually increase the predation of the fall armyworm, which then reduces the damage. Some crops also had very strong volatiles, like in this case, velvet bean or mucuna, a green manure cover crop that was very strong in repelling the fall armyworm, that also reduced the fall armyworm damage. So, and then lastly, we also had a specific situation in Southern Africa. We have a 7-month dry period from about May to November. And farmers grow maize under irrigation in such areas and really suffered from fall armyworm because there's nothing else green. The fall armyworm goes for those plantations, for those green maize production areas. And they really said, well, we have damages of 50–80% in our maize and we can't sell our green maize anymore. So what we did, we put a few lures in those and basically trapped all fall armyworm females in those. And And where we did that, we could control the damage in those plantations. And that's probably a strategy as well that I mean our colleague from ISIPI has now talked about. So in conclusion, several agronomic practices appear to influence fall armyworm infestation in smallholder conditions. Legume intercropping or strip cropping has been very beneficial. Conservation agriculture has been super effective. And organic amendments as well, because they increase the abundance of natural enemies. Those natural enemies, like the ants that I was talking about, help naturally control the fall armyworm. Some maize varieties appear to be more susceptible. We learned that from Prasanna no more in detail. Now we are a few years later. We have these tolerant varieties now that help, of course, in reducing. So combinations of the cultural practices and the good new elite varieties will really help us reduce those damages. And these technologies are in reach of smallholder farmers. There is a certain effect of seasonality. Some years were very dry, some very wet, and we found that this really makes a huge difference for smallholder farming areas and farmers' fields. In drought years, we had huge damage problems in some areas. Early planting is important. Frequent weeding as well, because often grasses in the stand are of the same nature. They're also cereals, and that's why fall armyworm can continue breeding on those. So if you weed continuously and have clean fields, you can control fall armyworm. Then crop diversification, as I said, is in any form beneficial because it will increase the diversity of not only one type of environment in agroecology. It brings in a whole range of new insects of various nature. And if nature is in balance, this is the best way to control an environmental pest like fall armyworm. Then finally, yield losses are seasonal, with perhaps an interaction with dry season, late planting, and high damage and yield loss. But this is really dependent on the seasonal situation that you are in and what we can expect from different types of seasons. So in retrospect, 4 or 5 years after the emergence, or it's even more than that now, it's 9 years now, we have adapted to the situation. We are not there like we are in Latin America, that everything is in balance, but we have new technologies, we have new varieties, we have intercropping strategies, and together they can work very efficiently in combination to control this damage, the damage of the fall armyworm. Thank you so much.
Thanks for your presentation, Dr. Christian, for sharing with us those interesting insights from your research. Okay, now we can proceed with Dr. Samira's presentation. Dr. Samira, are you able now to share your screen?
Presentation itself, it got corrupted. So now we are sharing.
Yes, we can hear you, Dr. Samira. You can try
Yes,
sharing your
yes.
screen.
Yeah,
Thank
we're
you.
sharing. Okay. You can see my presentation?
Yeah,
Yes.
go ahead.
Okay.
Can you
Okay.
try it in full screen, please?
Yeah, yeah, good, go ahead.
Yeah, all right. Thank you.
Okay. Okay. Thank you so much. Thanks, Bersana, for inviting me for this webinar. Thanks so much, much appreciated. And sorry for the technical problem. The presentation at the very last moment got corrupted, but I hope now it's fine. Okay. I am going to give a presentation on Berestoid, in particular focus on what we have done in East Africa, rather. Next, it's moving. Next. Okay. Okay, I think all of us, we know how probably unlike to many other continents, African continent is very vulnerable to invasion, and this is due to so many factors. And of course, always our the first response will be excessive use of insecticides, and it was also the case for the fall armyworm. So when fall armyworm it came, of course it came to find the environment is very suitable. There are so many host range there. And of course, as the previous presenter mentioned, it has high fecundity and high ability to disperse. And of course, when it comes to natural enemy, always when a new invader come, largely it come without natural enemy. But this is a yes and a no in case of the fall armyworm, and I explain further as we go ahead. So at ECB, as probably Subi mentioned, we have developed a comprehensive management for fall armyworm that include biopesticide, which my colleague Dr. Subi elaborated very much on this one, and of course we also optimize the trapping. And the good thing, by then we have a well-established boost system that was developed for management of stem borer, striga, and of course for soil health. And it happened when it was tested, it worked very well for this one. But here our focus on the parastoid. So there's some work that did by Mark Kenneth, and he realized that there are so many. The diversity of Africa is so high. Africa and other places where it's invaded, the diversity is very high. So we zero in to find out, and this could be largely this could be why the fall armyworm initially it was exploded, but after few time somehow it is stabilizing. We are not saying it's not a problem, it is still a problem, but it's not as it was initially when it came. And the parastoid could be one of the contributor to this stabilization. So we went ahead to find out what could be among this very high diversity, what could be the parastoid that form new association with fall armyworm, and we find several egg parastoid that Telenomus, which even though it was not native to Africa, it has been imported earlier. So it formed new association with fall armyworm, different species also of Trichogramma and Clonus also. They form that particular one is a egg larva. Three of them are egg parastoid, and one is a egg larval parastoid. And also there is a rich guild of the larval parastoid that also established new association with fall armyworm. Of a particular interest is Courtesia acb. It's why we tested it was quite efficient, and this is why we took it for further investigation. So as I will show in my subsequent slide. So we took also this the egg larval parastoid and the egg parastoid. We tested them in that purely under lab condition. And their parasitic range as from 45 for Trichogramma up to 90 for Telenomus. While for the larval parastoid, as I mentioned, the most effective among them is Courtesia acb. And we did further tests to find out which particular host stage that is preferred for this particular species, that's Courtesia acb. Of course, this for so many things. First of all, to optimize the mass rearing, because you don't want to offer the parastoid the wrong host, otherwise your colony will always collapse. And again, for the targeted releases, you want to find out when exactly should you do your releases. And interestingly, we found the early instars, those are the most preferred and most suited for this parastoid. When it comes to forcing star, it is not suitable. First of all, it can it defends itself against physically against the burstoid, and there is a very minimal cases of the encapsulation whereby the immune system of the host it encapsulates the egg or the larva of the burstoid. We also want to see when it comes to release of this burstoid, where could be the suitable areas. And as you can see from the model, around the equator, and as you go southward, it's quite suitable. And this under current climatic scenario, and this scenario remains the same even under future climatic scenario, with the slightly of the high suitable area shrink a bit. Having just to parasitize is something, but we went further again with this parasitoid to find out the host species, because when you release, the parasitoid will encounter so many other hosts. Some of them they even co-evolve with this parasitoid. Like let's say Spodoptera luteralis, but there are other bees symbiotic in the system. Interestingly, we find this parasitoid, it preferred, it all the associated host that is Spodoptera luteralis, but almost equally same as fall armworm, while the other hosts, the stem borers, they are not preferred. And of course, this translated to whether the parasitoid is able to distinguish between the good parasitoid or not, and those one which are not. And the suitability for this parasitoid for opposition, it was high for fall armworm and its closely related relative, Spodoptera luteralis. And we went ahead and test how it affects the development duration, and number of cocoon form, and the number of closed wasp, and of course the sex ratio. And for all these parameters, the performance of this parasitoid was highest in fall armworm and Spodoptera luteralis, as evident from the table. Of course, except for the progeny, it is comparable across the host where the parasitoid were able to develop. Then we went ahead, and now we establish all these parameters. We went ahead and do argumentative releases for several of these parasitoids, the larval parasitoid and the egg parasitoid, and their different initiative. One of the project led by SUBI, another an EU and European Union funded project. We made releases all in so many countries in East and Southern Africa, and with a very promising result. But to make the impact more sustainable, we train private sectors, and in particular Dream Team Kenya, and they release over millions. And the farmers, they can access the parasitoid from them at a very affordable price. Borrowing from what we did on fruit fly, where we do argumentation, where we test something called argumentorium. Argumentorium is like a tent. In this case, we use a small cage made of tent in such a way that the emerged larvae, or the hatch larvae rather, will not be able to exit from that netting cage, while the parasitoid will be able to exit. So we will collect the larvae, put them in this particular cage in such a way that the parasitoid, when they come, they drown in water. And we found out this approach it can increase the parasitism so low, while the eggs, the hatching larvae, the mortality is 100%. This, of course, we did it on a very small scale, like 1 acre, but just it's something to be taken into consideration. Then, having tested the parasitoid with bush bull system, of course, we already the suppression trait of the companion crop, like this desmodium and the grasses, this is well established. But we also found that the volatile emitted by this plant, they recruit parasitoids that help further in suppression of this pest. And okay, with another initiative, we want to establish, okay, now we have released those parasitoids, how far they have gone and what will be their impact. So we release this parasitoid and we measure their dispersal from the central point, and just in one season, we found that they can disperse in to more than 1 kilometer in a season, with a precision of almost about 80%. This about the native natural animal or already which are in Africa, but okay, that so now we have this parasitoid. The project come and go. We need to train the natural system of how to initiate and maintain those parasitoids, and we have done a very thorough training in Tanzania, in Uganda, and of course, as I mentioned, the plant village in Kenya. But being the invasive pest, of course, you agree with me, still the ideal target for the controlling it will be classical biological control. We are lucky IITA, they have already introduced 2 parasitoids. One of them is the egg parasitoid, and one of them is the larval parasitoid. So they share with us this material, and we test them in the lab. First of all, to be able to release them, we need to meet the requirement by our regulatory body in Kenya. One of them is to test their whether first of all they are effective against the fall armworm, and whether also they will attack other related pest or other related species. For the egg parasitoid, that Colonos insularis, it was quite specific, and also it is quite efficient. For the larval parasitoid, that's Cortesia maglomeritilis, it had a slightly broader host range, but it is even more efficient than the egg parasitoid. And now jointly with CIMMYT, with a project funded with CIMMYT, which just has concluded, thanks to Brasana and his team for the resource, we released this parasitoid in 2 countries in Kenya as per the directive we are given from CAREFIS that we have to restrict for now our releases. So we release in 2 countries, that's Machako and Imbo. And of course, before we did the releases, we have a baseline information of parasitism. So the parasitism by other is quite low. And then we did after the assessment, after the first release, after the second release, and as you can see, the release is increasing, but still this is just preliminary, and the releases we have done, we need to continue. We have just done a few releases, but this one need to continue if we have to have an impactful suppression for fall armworm. And I think there's so much we can do together. And with that, I want to stop here. Over to you.
All
Chairman.
right, thank you very much, Dr. Samir, for the presentation. So we're done with all of the 5 presentations, and I think our participants are very eager to ask questions. And in fact, some of them have already thrown in their questions in the Q&A chat box of Zoom, and some of our research speakers have actually started responding to them. But for an open forum for about 10 minutes, I now invite Dr. Prasanna to please kindly moderate our Q&A part. Thank you.
Thank you. I think, colleagues, you would agree with me that these are high-quality presentations on eco-friendly approaches to fall armyworm management. And many questions have come in, and which we are already responded to many of them, and some are being responded to by my colleagues. But if you have any specific relevant question that you would like to address or ask please raise your hand, and we will try and answer that. Okay, go ahead, and colleagues may raise their hand. And then Sunny, please enable to unmute them so that they can ask the question.
We have a question from Mr. Hin.
Yeah, thank you so much. Can you hear me, everyone?
Yes, we can hear you.
Yeah, thank you, Mr. Moderator. It is so exciting to listen to different research results based on the study on fall armyworm. I have a question for Dr. Christine. Okay, so my
Yeah.
question is about the presence of ants. What I heard from Dr. Christian is that ants are the best predator to deal with fall armyworms. So can they be bad for the crop as well? When we talk about ants, can they damage crop roots? Okay, thank you.
You have to
That's an
unmute.
excellent question. Yeah, that's an excellent question. And we are not talking of termites in this case. We are calling of real ants that forage around. And we've also found, interestingly, some of the legumes attracted some alternative prey, aphids, for example, which are not so good. We know that, but because aphids exudate some sugary solutions that attracted the ants, and then when the ants were there, they were starting to control the fall armyworm. So building on that, we put in some sugar solution as well in the fields to just test if that would make any difference if there was some sugar solution there. And indeed, that attracted again the ants, which then controlled the fall armyworm. So it's a bit complex because it's not a direct link. It's often indirect. What attracted the ants to come in and then see those fall armyworms and then control them on the sideline as well.
Subi, would like to add anything, Subi?
Yeah, I think Christian covered a lot. In our experience also, when we sprayed things like the fish soup, which is actually an animal protein, and ants goes for animal proteins, and specifically when they don't find, they start feeding on following. That's what we also realized as the management mechanism when you use fish soup, which very well relates to what Christian mentioned. Thank you.
Yeah.
Okay.
So
Any
can I
other
have a
question?
follow-up question? Yeah, can I
Yes,
have a
go
follow-up
ahead.
question? Yeah, thank you so much, Dr. Christian, for your response. Actually, your research is basically on corn, but armyworms, I can say damage a lot of crops in Cambodia, especially rice. So can you tell us a little bit more about how can armyworm become an outbreak? What are the best factors to cause an outbreak of an armyworm? Okay.
Well, I must say this is beyond my knowledge level, and I could not give you a very good answer on that because I have not worked on rice in the past. But maybe Prasanna, you have some responses that are more elaborate than mine.
About what? What is that question?
How to
I
prevent
just want to know
fall
the
armyworm?
effect of armyworms on rice because
Aha.
it is the worst case in Cambodia. Thank you.
Okay. So far we have not noticed any major economic damage to rice or wheat because of fall armyworm. So laying of eggs on any crop plant is not an indication it is causing an economic damage. The economic damage is something that one need to very carefully monitor. But as of today, there are no such reports. But one has to clearly watch out for evolution of the insect strains. And if there is any new acquisition of capacity to cause damage, one needs to carefully monitor. But as of now, no.
Okay, thank you. Thank you.
So,
Thank you.
so can I actually have some?
Yeah, please.
So, in our in our country, Bangladesh, in the Bangladesh Rice Research Institute, we did some these laboratory works on this fall armyworm on rice, and we have seen that fall armyworm can develop on rice. Maybe it takes some more times than the maize, but in the fields we are not getting any infestation. The main reason is behind that because rice is generally needs more water, and one of the lifestyles of these types of armyworm, like this Spodoptera litura or Spodoptera this progipada, this pupation is actually going for pupation underneath the soil. So as there is this muddy water, and due to that, we have seen even Spodoptera litura or Spodoptera fijupada cannot actually sustain in this rice field. So this is the main problem, but we are suspecting in some of the this upper land rice like this in our country, house varieties or something like that, where less water is using, might be a chance of infestations, more infestation of fall armyworm. Thank you.
Yes, thank you. I'm sorry that my question is out of the topic today, but I just don't want to focus on rice. I'm curious about the outbreak of fall armyworms. In case we talk about corn again, have you observed any specific factors or situations that can cause an outbreak of fall armyworms? Yeah. Even we talk about corn. Thank you.
So, Dr. Dr. Alam, have you seen any observation of fall armyworm in rice in Bangladesh?
No, not in Bangladesh, but there was some probably from Tamil Nadu. There was some infestation we have seen, but it's not so much. But in Bangladesh, not in the field. But as I told you, in the laboratory, we have seen that they can actually all the four this life cycle stages can be actually okay in rice. They can actually do it, but it takes some more times. So there is a chance. And as you know, there is two strains of this fall armyworm. One is rice strain. So if there is the rice strain can attack, so there may be also a chance of more infestation.
Okay. So
yeah,
yes,
I just want to quickly add on to this. In this case, for a pest outbreak, there are a lot of factors that contributes to the outbreak. Especially in an irrigated situation. Rice is an irrigated crop, and if in an irrigated situation you don't have anything else around, following a maize season or something, yes, the rice in that irrigated farm can get damaged by fall armyworm. This has been seen with maize as well. Irrigated maize is much more susceptible to fall armyworm as compared to any other because of their presence alone. So such kind of situations can happen, but wide reports on fall armyworm damaging rice has not come up so far, even in the presence of rice strains and the maize strains as well. Yeah, thank you.
Yeah, thank you.
Okay, so any other question, please? It appears most people are happy writing those questions on Q&A, and we are trying to respond as much as possible quickly. Okay, if there are no further questions, no raising of hands, thank you so much. Sunny, please go ahead.
Thank you very much, Dr. Prasanna, and to our resource speakers for graciously responding to the questions of our attendees. All right, now, so before we close, we just have some few more things. The evaluation form. We'd like to hear your feedback to our webinar. This is also our certificate request form, so kindly fill out the form, type in bit.ly/54solve, or kindly scan the QR code for you to access our feedback form for today's webinar. Okay, I think that's it.
Okay, with that, thank you so much. And once again, our heartfelt appreciation to the CRCA team, including the director, Dr. Nova, as well as Sunny, and the entire team, which has put forward an excellent webinar structure together. and to enable us to reach out to all of you. And my sincere gratitude also to all my colleagues, the resource persons who have spared their time despite their schedules, especially colleagues from ISIPE, Dr. Subi Savagan, who is always there. We have done perhaps this many, many times together. and Samira, who is another fantastic scientist. Both of them have always been there to support this kind of knowledge sharing events. And to my own colleague from Christian Terpillo, he is an outstanding cropping system specialist and one who loves to work with the farmers. And that passion one can see in his presentation again today. And thank you once
Thank you
again,
so much.
Christian, for joining. And last but not the least, our very dear friend from Bangladesh, a very renowned entomologist, a former CIMIT consultant and ex-director of Bangladesh Agriculture Research Institute, Dr. Syed Nurul Alam. Again, he is someone who is passionate about this field. And whenever we requested him to give any talk, he never even hesitated a second. So Dr. Alam, very, very grateful to you for always sparing your valuable time. And I'm sure that we can do a lot more in the years to come, thanks to your passion. So my participants, I can see more than 150, 60 participants, 160 participants, very actively engaging and asking questions. What is really important is don't consider this as a one-off event. You have our email details. Sierka can provide them along with these presentations. If you have any follow-up action that needs to be done, whether it is with regard to access to different technologies, or whether it is regard to any future possibilities of collaboration with organizations like CIMIT or from like ISIPE, don't hesitate to reach out to us. Sierka is a fantastic facilitator, and we can do many, many things together. I must also gratefully acknowledge the support from Melinda. Melinda Limlenko is my colleague who has formed this bridge between Sierka and CIMIT and enabled organization of this workshop. Many thanks, Melinda, in case you are not sleeping, but I know it is it's almost early hours in Mexico. Thank you so much, friends. We really enjoyed participating in it, and I'm sure Sierka will follow up with many other follow-up actions. Once again, thanks a lot. Take care.
Thank you,
Thank you.
everyone.
Thank
Thank you to all our
Thank you.
attendees. And of course, to
Thank
our international experts, thanks everyone. And of course, on behalf of Sierka, our sincere thanks to CIMIT as well for this wonderful collaboration to bring fall armyworm management knowledge to our audience here in Southeast Asia and even beyond the region. And to our attendees, we hope to see you in the next webinar. So please stay tuned for announcements on Sierka's social media channels. Again, my name is Sunny on behalf of Sierka. Maraming salamat po. Thank you very much. And we wish everyone all a good day. Bye-bye.
Thank you.
Bye-bye.
Bye-bye.
Thanks, Dr. Prasanna.
Bye-bye.
Bye.
Thank you.
Come, let's build the center that we'd like to be. A place where you and me can work in harmony, where everyone is proud to serve with dignity. Come, let's make this dream a reality. Fill our hearts and minds with a vision. Let our words below our mission. Leave our values on excellence in work, commitment and unity. Come, we have a stream for the senior region, extended to the world throughout generations. Fill our hearts and minds with ambition. Side by side we stand for one mission. Leave our values on excellence in work, commitment and unity. Let's show the world we can. With God close at hand, we will grow together for the better.